All right, so we are back. We're going to look at the third, I guess, final video that we're going to kind of take a look at with the biomolecule or with the biochem unit. And we're going to kind of cover on this one, we're going to plan on going over is the four major biomolecules that are utilized, needed in order for us to function properly, right, correctly. And again, when we're talking the four major ones, this is not the only ones, but the ones that probably have the largest impact on us. And so I'm just going to kind of start a little bit by talking about the overall process of these biomolecules and obviously them being built up from atoms and the bonds that are happening between them, right, which can bring on some special characteristics that we'll talk about a little bit, right. Um, and I think probably more importantly, as we take a look at the big picture, we're starting to take into consideration, these are ultimately they're polymers, which means that they're made up of building blocks. They're made up of very small subunits, and just the overall process of how our body utilizes them. And so, kind of in the first step is that in order for us to build up the molecules and eventually become us, is that we have to take in these molecules, we've got to break them down, and then eventually build them up into something that we can utilize and that we need. Obviously, they all have their own very important function that we want to cover. We want to talk about their structure. Can you actually acknowledge their structure and maybe some of the characteristics that come from that? And just basically any of the other kind of odds and ends as far as why and how we utilize them and whatever just kind of weird, I guess, quirky little things else that we need to know about them. First and foremost, in the process of breaking monomers, or sorry, building up monomers and polymers, and then also breaking polymers back down to monomers, there's a couple steps that are occurring within that process itself, and that we're talking dehydration and hydrolysis. So if you guys remember, we talked about this at the beginning of the class, beginning of the school year, is that when we start looking at this reaction right over here, where we're taking two monomers, two amino acids, and we are basically bonding them together, right, to form a polymer that we see over here on the right-hand side. There's something that's occurring, right, within the carboxyl group and the amine group that we find on each of those amino acids. And in order for this reaction to occur, right, we can see that there is a hydroxyl group and a hydrogen group that are released, right, from each of those amino acids. And due to that, those two eventually will form together, and they're going to form water in the process that we're going to call dehydration. Dehydration, again, because water is given off. Right? If we want to basically reverse this process and break that polymer down into monomers, right? Again, water is needed to get those molecules back to their original state. And so again, therefore, we're going to call it hydrolysis because water is needed, water is required in order for that to happen. So every time that monomers are built up into polymers, dehydration is occurring. Every time that we're breaking those polymers down into monomers, hydrolysis is occurring at that point in time. Okay, so we're going to start with the first one, start with carbohydrates. Obviously, a most important factor of carbohydrates is its ability to give us immediate sources of energy. When we start looking at them versus fats give us the most bang for their buck, but fats are used more for storage. These here, immediate source. And again, even then we can break that down into, how quick we get that energy just based on it being a monomer or polymer. As we start breaking down the monomer form of this, we're looking at basically a monosaccharide, saccharide meaning sugar, right? The mono obviously referring to one. And so our body, what we want to end up doing is that if we could take in carbohydrates, such as a polysaccharide, which would be a polymer form of that, is our body needs to break that down by the process of digestion or also using enzymes, which we'll talk about proteins here in a little bit, right? And it's going to break that polymer down into the monomer form, which then we can use either for immediate source of energy in the process of cell respiration, so on down the line, which we'll hammer out in another video coming up. So you kind of got the whole polysaccharide versus monosaccharide form of the energy. There are some carbs, remember, that we cannot break down, fiber, for example, and we did go through in our notes, and we talked a little bit about the enzymes that we do not have that basically do not allow us to break down the specific shape of fiber. It's all based on the fiber itself and the shape of the fiber and its molecular makeup and why we can't break it down, where herbivores can. And so again, you kind of start looking at again that overall theme of shape determines the function, and the structure and the function and so on, and how that can manipulate or basically kind of change or alter the way that we are surviving, when we can use things. But fiber is obviously very important to us because, again, it is going to help to keep us regular and push food along, right? The other part that we kind of started talking about a little bit with carbohydrates was more along the lines of, storage and just the overall process of our body's ability, where can we produce, where can we actually store some of this excess amount of carbohydrates. Remember that we do have in our body about a 24-hour supply that we're going to find inside the liver. It's called glycogen, that any point in time that we need. I've always used the example of kind of like Fruit Ninja, that if we need to kind of slice and dice a little bit off here and there and in a fight or flight type of response, we have that ability to do that if we need to. Plants are a little bit different in how they basically will store theirs. Amylose, amylopectin, more in that starchy kind of format, which is why when we take in plants, we are utilizing that starch that they are ultimately storing up inside of their cells. So we kind of got that going on. We start looking at maybe some other things. Chitin is an example of carbohydrates that maybe would come up. Some organisms, again, are going to need microorganisms to break it down. Bacteria, basically termites are going to have that kind of mutualistic relationship with protists that they find to help them break down the wood that they're taking in. And so, again, just a lot of different scenarios of dealing with the carbohydrates. But again, for us, we're talking primarily energy, right? Okay. Now we start looking at lipids. Again, if we're talking about energy, we're talking about the most bang for its buck, right? We're talking about a source of energy that we can't break down very well, right? One, because it is a non-polar molecule. And so therefore, because it provides with a lot of energy and because we can't break it down very easy, right? Well, what do? We're going to store it. We're going to basically save it for a rainy day type of situation. The one thing about the lipids, there really is no true monomer. The one thing that we kind of found out with this group, or that they've determined with this group, is that the major characteristic of all lipids is that they just don't mix poorly, or they mix poorly with water, meaning that they're non-polar. So you take a look at a fat, for example, obviously doesn't mix. Okay, you take a look at the phospholipid bilayer of the cell membrane. Okay, doesn't mix. Steroids. Or another example of that, hormones. There's some hormones that are, which we get back into the cell signaling here in the next couple of videos, right? We'll bring that into play again. So there's a lot of molecules in our body that simply are just non-polar and they don't mix well with water, which obviously is very advantageous for them. Remember, we talk about fats. Okay, we talked first and foremost about saturated versus unsaturated fats, single versus double bond. Animal generally versus plant doesn't always hold true. Again, there's always an exception to the rule with most things that you're going to learn in biology. And so we've got kind of got that going on as far as the two different types. We broke down a little bit as far as, well, why plants maybe don't have fats, why we do. And again, you kind of look at our ability to move, locomotion, the type of energy that we require in order for us to survive versus plants. I mean, there's just a lot of complexity issues there as far as maybe why plants don't hold theirs in the case of lipids, and why we basically are kind of stockpiling this energy up a little bit. But anyway, lipids very, very important when we start looking at just the amount of energy that they can provide for us. And cholesterol, again, we did talk a little about cholesterol and its advantages, and that basically it's the main precursor to steroids. So yes, cholesterol is good, just in moderation. Obviously, too much cholesterol leads to heart diseases, arteriosclerosis, and so on down the line. So you've got the lipids going on. Probably the biggest thing that we start looking at with the biomolecules, I think, that is fresh in your guys' mind, though, are proteins. And we start looking at not really just what they do, because we've learned that they virtually do everything for us. You name it, inside of a body, they're probably going to have some function. They're going to have something to do with it. And probably the biggest, we always want to break down the muscle tissue and building up muscle, but really we're looking at enzymes, and we're looking at the body's ability to break things down and build things up. Well, it's all due to proteins, enzymes that our body produces or makes. We also go back to our traits. You are you because of the proteins that are being made. And so I think when we kind of look at the proteins in its monomer form of an amino acid, I think probably the biggest thing that we really need to take into consideration, or really kind of have our mind wrapped around, is just the overall building up of a protein. And as we've talked in the past, and we'll go to this slide right here, is that sometimes, these proteins can be anywhere from basically what we kind of relate to a strand of yarn, right, to something that can be a complete sweater. So some very, very simple, some very complex, but no matter what, we're looking at a building block of an amino acid. And again, the amino acids are basically all built the same except for one thing, and that's the little R group that we find right here, that side chain, right? And that is what gives each amino acid its ability or its characteristic that it does. It can make it nonpolar. It can make it polar. It can make it acidic. It can make it basic. It can make it interact as a hydrophobic kind of molecule, or hydrophilic. It can make it form disulfide bonds with other molecules. And there's just so many different characteristics that this side chain can ultimately make that amino acid, which eventually will give that overall polypeptide or that peptide chain different characteristics as well. So we're talking bonds. Okay, we're talking peptide bonds specifically that are going to bind amino acids together. And when we break these down, right, we're going to break these down into basically the 4 different levels, right? And to, again, just having that idea, that overall thought process of what's occurring at each of these levels. And so in number 1, right, we're looking at amino acids, right, basically are just going to be held together by peptide bonds. And so when you go back to remember how are amino acids, how do they know when to pair up, where to pair up? Well, it's all happened inside the ribosome, right? And the ribosome ultimately is getting a set of instructions from the mRNA, which is getting its instructions from the DNA. So every enzyme out there, every protein that is being built is all dictated by DNA, right, that we find inside of our cells. Okay, so keep that in mind. So we go to level 2, things are happening a little bit different in that we're going to find hydrogen bonds are going to occur, right? Basically due to interactions between hydrogen and oxygen. Nitrogen can get thrown in there as well with it because it does also have a high electronegativity number. So you're going to find some folding that are going to start to occur, and it'll either cause it to be more of a spiral shaped or a pleated shape, just depending on those interactions. 3, more folding is going to occur, but now we start finding more of the interactions between the R chains or the side groups to where disulfide bonds can form, hydrogen bonds, hydrophobic, ionic bonds, all those different types. And now we really start to see that molecule, as you see up here in the right-hand corner, really start to turn back over each other. And then if it does ever get to level 4, right, we can start to see that these proteins, right, the polypeptides are now going to start to bond together. And so all coming back to the amino acid and the side chains that it has, this shape right here is going to be something completely different for each protein. And as we've talked, one of the major themes, structure determines a function, right? And so again, whatever structure it takes on, it's going to give it a brand new function. All right. Now we'll kind of jump into enzymes a little bit more as we go on because we'll talk about a little bit more about the substrates, and I'll probably start that out with my next video as we go through. We'll talk about the substrate and what's being broken down and the concentration of the enzymes and what that does to the overall reactions. We'll talk about how they become denatured and how this overall molecule can change its shape, right? How the active site and what they kind of do for it. But again, just the overall process of the protein is going to be very important as well. So muscle structure, traits throughout the body, any enzyme that we basically find is all made from protein, which is ultimately determined by the DNA that we find inside of our body. So again, as we go through these, anything that you guys want me to make sure that we are cover a little bit more specific, shoot me an email, all right? But again, just quick little brief course over BioChem, right? And again, I hope things are going well with you guys. We'll probably talk to you a little bit more on Friday morning. We start going over those long kind of the FRQ questions on the review packet that I gave you. I'll see you guys.